Would any of this be possible if we didn't want to have sex? Why do we want to have sex? And that's kind of what I'm going to explore today. How is sex in the brain acting like a nuclear fusion generator that takes the pleasure that we experience from it and generates desire from that? Desire for partners, desire for features, desire for that state, that beautiful state of incandescence that happens when we feel like sexual beings. So here's some food for thought. When you think about why we have sex, there's really two things that come to mind. Gislin says, we've evolved a nervous system that acts in the interest of our gonads and want to tune to the demands of reproductive competition. Now, in a sense, that means that everything is set in stone. Those who compete and win spread their genes through the gene pool, get represented more in the gene pool, and we should all, if that's true, have breasts the size of our heads, hips the size of 3 chair lengths, and members of my gender should be big enough to wrestle bears, would tear the meat off with their teeth. Why hasn't that happened? Because there's another reason why we have sex. This is written by Krafft-Ebing almost a century ago. Krafft-Ebing said erotic fetishism makes an idol of physical or mental qualities of a person or even merely of objects because they awaken mighty associations with sexual pleasure. The idea here is that reproduction is a fringe benefit. You have sex simply because it feels good, and it makes you feel good, or the other person makes you feel good. Well, to study this, how do we do that? Obviously, the best thing to do is to study human sexuality in humans. Problem is, there's not a lot you can do studying humans. Masters and Johnson were very lucky to take undergraduates and pair them up together and get them to have sex and watch them having sex and come up with a human sexual response, a physiological human sexual response that goes through excitement to plateau to orgasm and then a delicious afterglow of the resolution where you lie together in facial closeness and fall in love, or where you get up and go. But it's an end point, okay? Others have studied desire in women related to the point of the ovulatory cycle. And sure enough, women have more desire around the time of ovulation. We think we're freed from the cyclic tyranny of our hormones, but we're really not. But you can have sex every day. But when you initiate, as a woman, having sex, well, there's a point at ovulation, and this pointer doesn't work. But you can see it here. The point at which estrogen and progesterone have primed the system to link it to reproduction and to increase the chances. And we can look at the effects of hormone replacement in hypogonadal men and see that as you increase the amount of testosterone that they get, sure enough, they get more erections, they get more nocturnal erections, they attempt to have sex more often, they masturbate more often, and they can have orgasm more often. And that's all well and good. And of course, in the last 10 years, the advent of Viagra and other phosphodiesterase 5 inhibitors have given us insights into sexual desire in men and women. But when we think of desire, we think of an intractable chemistry, something that happens that makes us, without any thought of the consequences, engage in sexual activity. In order to understand that, we've had to make use of animal models, because I can't study the brain in a human. I can't take it out. I can't look at what's being activated. So we make use of the fact that really all sex has the same beginning, the same middle, and the same end. Whether it's an obnoxious human or I don't know what the icky is, but maybe it is icky, who knows, in that species. But we take advantage of the fact that sex has a beginning, a middle, and an end, and that it's subserved by a brain that Mother Nature in her infinite wisdom says, if this system works, if this transmitter, neurochemical system works, it's a keeper. We're not a Monty Python skit. It's not something now for something completely different. Mother Nature looks at the lower and says, that transcends evolution. So I study sex in these guys, the rat, the lowly rat, who, when they're having sex, ain't so lowly. This is the best porn shot of rat sex I've ever seen in my entire life. Why use rats? Well, it turns out rats are a lot like we are. There's a large scientific literature, so you're not groping in the dark for a phenomenon. They're small, so you can test many at once, and you can watch them. You can watch them have sex, which is what you want to do with people, and you can't. They're highly social animals, right? Just like us. There's a high degree of behavioral and environmental plasticity, just like us. And their sexual behavior, which is described quite often as being polygamous, is, as you will see, highly opportunistic and highly malleable by their experience. Here's what rats do. There's an left-hand side. There's a female. She's on the ledge below. She's greeting a male who's coming down. She hops over him, exposing her anogenital area so he can sniff and lick. Then she kicks him in the face and runs away. That's a solicitation. A little bit grade 5, but it's a solicitation. She runs away, she goes down, she holds this arching of the back called lordosis. That allows the male to intromit, get his penis in there. He pulls out, he grooms, she runs away. She comes back and does this several times. He intromits several times until he ejaculates. Of course, then he goes to sleep. But he learns during that sleepy period, as you will see. But during that period as well, he recoups. So he can ejaculate up to 10 times before he's completely sexually exhausted. So when we think of rat sex or animal sex, what are we thinking about? What is the role of learning? Aren't they just Nike commercials? Don't they just do it? No, they learn about their desire. They learn about their arousal. They learn to overcome obstacles to sex or mating. They learn to locate mates. They show mate recognition, courtship behaviors, copulatory behaviors. I mean, if you ever watch rats copulate the very first time, you'll wonder why there are so many rats. They're stupid. But they get it together. They learn how to do it. And they learn sexual partner preferences. And this is where we're going to take off. So where does desire come from? Well, I'll cut to the chase. It comes from Woody Allen's second favorite organ, his brain. In order for us to figure that out, we have to ask rats human questions and disguise them so that the rat can give us the answer in rat terms. And we do this using chambers that allow the female to regulate sexual contact. That's what she likes about sex. What does the male like about sex? Well, it turns out he likes to ejaculate. This is a pacing chamber, bisected by a plexiglass screen. The screen has holes in it. The female can run back and forth. The male can't because the hole's too small. So, of course, he's left there waiting, hoping, praying that she'll come back. We looked at things like sexually conditioned place preference. So they associate one side with sexual activity to ejaculation, maybe the other side with sexual activity but not to ejaculation. And what do we find out? Well, in male rats, ejaculation is necessary for that place conditioning, for them to associate the cue of the place. They don't find this in their real world, but they're able to do that. Their brain is able to sustain this kind of learning, only if the male ejaculates. Furthermore, there's stronger conditioned place preference when the males are more aroused prior to ejaculation, as if humans didn't know this. And the acquisition of place preference is blocked by drugs that antagonize brain opioid systems. You've heard of the endorphins and the enkephalins. These are the systems that produce pleasure in the brain. You block the pleasure, you block the activation of that learning. But it's not blocked by dopamine antagonists. We've heard a lot about dopamine today in the talks about dopamine and reward, dopamine and attention to reward. Well, it turns out that this is something that's very easy to learn. You don't need a hell of a lot of dopamine to figure it out. In female rats, a very similar picture emerges. The ability to pace the initiation and rate of copulation is necessary for the conditioning. So females like it on their terms. Men, are you listening? Ladies, are you listening? Females like it on their terms. Females prefer a 4-hole divider in that pacing chamber as opposed to 1-hole, mostly because in the 1-hole, the male puts his head in there like a cork in a wine bottle, and she's got to play basketball with his head to get it out. 4-hole, she directs in one way, goes in the other. It was ever thus. Females, as Vivian said, are the ones who have to pick up the slack. We can induce timed clitoral or vagocervical stimulation that's capable of inducing place preference. And the acquisition is blocked by the opioid antagonist naloxone, but not by dopamine antagonists. Now, this is astonishing. What we're seeing here is that the brain of a male and a female rat are essentially the same. The pleasure is essentially the same. Where's the great sex difference? Maybe it's not really there. We look at sexually conditioned partner preference in my lab, where we use an odor like almond. It means nothing to a rat. You can make it bad by making them sick, or you can make it good by putting it on their partner. I think the perfume industry has known about this for a long time. We test them as little dyads, but then we train them as dyads, but then we test them in a ménage à trois, where the male on top or the female on the bottom has access to two partners, one that's scented, one that isn't. Now, if rats are polygamous and they've only ever had a scented partner, what should they want to do? They should want to go after the unscented one to spread the genes far and wide. Gee, do they do that? My students and I have examined this in males and females. These are some of my students who've done the work. Todd Kippen began this about 10 years ago looking at olfactory conditioning of sexual behavior. So we had the almond-scented receptive group, that's the paired train, so the training is paired with the odor. Non-paired trained get unscented receptives. There's also the unpaired trained who get access to the almond alone without it being related to sex, and then unscented copulation test trials. And then on the final ménage à trois test, they get the potential of two. Who do they do? This is our question. Well, it turns out it's very interesting. If you look at the bottom here, you've got paired trained males, non-paired trained males, and unpaired trained males. And what you have is the proportion of males who choose either the scented or the unscented female for their first mount, their first intromission, or their first ejaculation. And what hits you like a ton of bricks is the fact that they are not showing polygamous behavior, they're showing elements of monogamous behavior. The paired trained males are ejaculating preferentially with the scented females, despite the fact that they're copulating randomly with whoever's in front of them, being good males. And it's not that the non-paired trained males don't do anything. If you look at the actual mean number of ejaculations, it's exactly the opposite. The paired trained males are ejaculating more, that is, they're showing a mate choice more with their familiar female, and the non-paired trained males are showing a mate choice with their familiar female. We always thought familiarity bred contempt. Here, it's breeding desire. Todd also found, if you look at the left-hand side, that the animals that were given different types of sexual stimulation showed a different effect. That is, intromissions alone, without ejaculation, did not support the learning. Ejaculation alone didn't support the learning. But spending time in the PEI, post-ejaculatory interval, is where they learned it. Now, this is crazy. When the male is asleep, he's learning about her. And this harkened back to Stendhal's principle from 1796 in De l'Amour, when Stendhal said if the couple, after the generative act, they couldn't call it what they should have called it back in that era, shows facial closeness during that afterglow, they will bond. If they don't, they won't. So our rats are telling us exactly that Stendhal was right, and probably right not only for our species. The cool thing about odor conditioning is you can take the odor and get sex out of the picture. So it's like watching a porno for rats. You can present them with the odor alone and see the activation of different brain areas. Alpha MSH, the melanocortins, these are desire peptides. We see the activation of them. We see the activation of oxytocin, that's the OT. We see the activation of dopamine, very selectively to the odor, which is now a priming cue. And you wonder why her wind song stays on your mind. The odor is a priming cue. When we give the odor, we find it, notice the top left-hand side, for the paired group, the odor induces whopping increases in dopamine release that are also produced by just receptive female odors when we take a vaginal swab. Lemon odor that's never been paired doesn't produce that effect. And in the unpaired group, the almond odor doesn't produce the effect because it's never been paired. So we have a Pavlovian conditioned response that's happening in the brain. Not only that, when we give the odor alone, we activate oxytocin neurons selectively in the paraventricular nucleus. That's the PVN up there, not in the SON. So oxytocin is not being selectively released by the pituitary, it's being released into the brain to subserve bonding. Whereas vasopressin, on the other hand, is being released by the SON into the pituitary to increase your heart rate, maybe produce a little bit of water retention. When we give oxytocin or vasopressin or saline to males on their first conditioning trial to see if we can advance the learning, and then give them their ménage à trois, what do we find? Sure enough, look at the OT versus the VP. The oxytocin pre-treatment produced conditioning. It enhanced conditioning. And this is very important because oxytocin is about to be released as a nasal spray to enhance erectogenic effects. You wonder whether this is also going to have some effects on bonding or love. What about females? Other students in my lab have contributed to this work. And essentially it's the same picture, but as Darwin would have suggested, it's more powerful. The females not only show, as you'll see, a choice for ejaculation, but they stay with the paired male. So look at the paired group. They solicit the scented male more often than the unpaired or random group. They hop and dart, which is kind of like a proximal solicitation with that male. They take his first mount and they give him more mounts. And astonishingly, they take his ejaculation and impose an interval between his ejaculation and a second ejaculation that essentially, for 100% of those animals, confers paternity to that male. Think about what that means for a second. Mate choice. Mate choice, not just, I'll have sex with you. Mate choice is now based on who did you the best. That makes us rethink some evolutionary theory. Not only that, when we give the odor, we activate the brain in these places that are associating all of these neurotransmitter effects. The medial preoptic area, nucleus accumbens, areas that are involved in desire, the MPOA, and reward in the accumbens. We see that the odor acts as a prime in these regions in the paired group, but not in the unpaired. Furthermore, as with conditioned place preference, when we give naloxone in males, we block the development of the conditioned partner preference. And interestingly, males that are treated with naloxone during the training, they're not given naloxone on the final test, do not pursue females or work to obtain access to copulation. These are men, or males, excuse me, I sometimes get rats and humans mixed up in this. These are males with a sexual desire disorder. When you remove reward or pleasure from sex, why bother having it? And that's what the males are doing. They're saying, you go away, you come back, I don't care. What about the females? Exactly the same thing. Naloxone pretreatment during training blocked the development of the partner preference, and interestingly, females that had been treated with naloxone as opposed to saline during training, they don't solicit the males. They show reduced lordosis. They receive fewer mounts, fewer intermissions, fewer interactions with the males. These are females that, despite the fact that they're primed fully with estrogen and progesterone, simply don't want to have sex anymore because it didn't feel good. What about dopamine? We gave an antagonist called flupentixol. During training, it did not block the development of conditioned partner preferences in males. But, nevertheless, the males treated with flupentixol during training didn't pursue females or work to obtain access to copulation. So dopamine and attention are involved too. And in females, on the other hand, flupentixol did block the development of conditioned partner preference, and like naloxone treatment, it produced females with a desire disorder. Okay? So we have reward and attention to reward having effects in almost virtually the same direction. So in summary, activation of brain opioid systems during sex, but especially after orgasm, induces a state of sexual reward or pleasure. Activation of dopamine systems before and during sex allows animals to attend to reward-related cues and then to go after those cues. Activation of oxytocin systems before and during sex promotes bonding to reward-related cues. So essentially, these things happen all at once. You're not thinking about them. You can't think about them at the time you're doing it, or else you're not doing it. You're thinking about it. But Kraft-Ebing was right. Experience matters, and in particular, experience with reward matters. And what we're finding with sex now dovetails nicely on the work that people are doing with drug addiction. We're finding that pathways for sexual reward and incentive motivation or incentive responding overlap with pathways for bonding. That opens up important new avenues for the study of drug addiction. Why is it that some individuals bond to their heroin or bond to their cocaine and others don't? Why is it that the guy you want so much to bond to you simply doesn't want to? Right? Why have we preserved diversity and individual differences in the activation of these systems and hence in our species? When we think of the repercussions of sex, don't just think babies. Think the path of Cupid's arrow. Here's Cupid. Who was Cupid's lover? Psyche. the mind. Cupid takes control. And what you see is the path of that arrow going through reward, attention, arousal, bonding, and desire. And really what that translates to in a very physical, reductionist sense is the activation of brain opioid systems at the very basis of sexual reward. The activation of noradrenaline and dopamine that give you arousal and attention and take the body and merge it with the mind so that you can be sexual. And also oxytocin, vasopressin, and the melanocortins that activate bonding and that all funnel it down to desire, to seeing that collection of features that you can't resist. And I'd like to acknowledge all the people in my lab, my teachers, my graduate students, my undergraduates, Concordia, my center, and CIHR and NSERC, and CONACYT of Mexico for funding the research done in my laboratory. This research involves a large number of people, and of course it involves a large number of rats who are probably some of the happiest rats in all of North America. Thank you very much.